ABO forward and reverse. Again, you're going to fill in the squares. So you have a blank table. I tell you the person is A with your anti-A, your anti-B, your A cells, and your B cells. If I'm an A, would it be positive or negative?
Positive.
Positive or negative?
Negative.
Positive or negative?
Negative.
Positive or negative?
Positive.
Okay, so you're simply going to fill in the chart. I give you the blood type, you're simply going to fill in the chart. That's all that question is telling you. So for your A, B, AB, and O. Know the antigens and antibodies associated with each ABO and O Bombay blood type. If I am a type A, what antigen do I have on my red cells?
A.
A antigen. What antibody do I have in my plasma? B. Antibody. So again, you're just going to fill in the chart. Make sense? B, you are B antigen, A antibody, right? Okay. So on your AB, what antigens do you have?
Both. A and B.
A and B. What antibodies do you have?
No.
Correct. Okay. All right, so that's the, again, just filling in the chart. There's 131 points on this test, and a lot of it is that simple. It's just filling that in. Okay. O Bombay blood type. What makes them different? What antigens do they have? O's have no A and no B, right? But what does Bombay not have that everybody else does? H. Exactly right. Know how and when ABO and RH antibodies appear in the plasma. ABO antibodies appear when? After about 3 months, right? Because they're not present at birth, right? Not your antibodies. Your RH. What causes your RH antibodies to show up in your plasma? Exposure. Exactly right. Exposure. Blood transfusion, pregnancy. You have to be exposed. Exactly right. Know the antigens and antibodies associated with the RH blood type. So if I have D antigen, what is my RH type?
Positive.
Positive. If I do not have the D antigen, what is my RH type?
Negative.
Correct. What are the percentages? Of 100 people, how many of them have the D antigen? 85. Exactly right. 85. Which leaves you 15 for the negatives. Number 5, how to recognize and categorize missing I'm sorry, number 5, okay. Missing or unexpected ABO discrepancies. So that's what we did on Wednesday. Pretty much exactly like that worksheet. Okay? It's missing or unexpected in the forward or the reverse. Match the gene responsible for the ABO terminal sugars. So remember your D-galactosamine, whatever's for the B, and the D one is for the whatever. So again, it's just matching those. Know how immunoglobulin know the immunoglobulin class, reaction temperature, and frequencies of A1, A2, B, O, and RH. What is the immunoglobulin class of A and B antibodies?
AGM.
IgM. IgM. When can you have IgG in the ABO blood group system?
Pregnant. Or blood transfusion.
When an O person is exposed, either through pregnancy, right? Okay. Reaction temperature for the ABO blood group system. What temperature are you doing the testing?
Room temp.
Room temp, exactly right. When you do the D,
Warm.
room temp initially, right? But then if it's weak D, you have to
Incubated.
incubate, exactly right. And the gene frequencies of A1, A2, remember I told you 80% of the population is A2, or I'm sorry, A1, 20% is A2. In the O blood group, and then the RH. So again, RH, your D, I already asked you, is 85%, negative is 15%. Know the Weiner-Fisher-Race nomenclature for RH antigens. I'm going to give you one, and you're simply going to match it to the other one. Just simple memorization. Okay. Why is the SE gene important to blood groups? What does the SE gene do in blood groups? If I have the SE, what am I called? Secretor, right?
Oh
So
yeah.
your SE gene determines if your antigens are where? In your saliva. So you can spit your blood type. You are a secretor, right? So again, big SE, or big SE, little SE. It's dominant. So in other words, if you were little SE, you are a non-secretor, right? Okay. Know the contents and characteristics of your blood bank reagent. So what is in anti-A?
And
Antibody. What is in anti-B? Antibody. What is in D? And
Keyword.
potentiator. Correct. What is in your RH control?
D antibody.
No,
Potentiator.
just the potentiator. Exactly right. A cells. What is in your reagent for your A cells? What's in that bottle? I'm sorry? A antigen. Absolutely. And in your B cells, what is in there? B antigen. Exactly right. Exactly right. So if you can remember, your cells are always your antigen, and your antisera is always your antibody.
I want to ask a question.
Sure.
You said D, antibody, and potentiator
Uh-huh.
for like anti.
Anti-D.
Okay.
And then what's in your RH control?
Potentiator.
Just the potentiator. Correct. What color is your anti-A reagent?
blue.
Blue. What color is your anti-B reagent? Yes. Okay, determine the possible genotypes and phenotypes of offspring given the genotypes of the parents. It's a Punnett square exactly like the last one. Okay? You have to remember in blood bank, everything is you gotta remember everything from day one all the way to the very end. It's not like chemistry, we only had to study proteins, okay? So it's cumulative. It's everything from day one all the way to the end. Because once we learn AVO, doesn't mean you get to forget it now that we're on RH. Alright? So, I have told students in the past, always go back and look at your old tests before your next test, because I repeat questions. Okay, again, I told you test 1 was the Punnett square, you're gonna see it again. So if you got it wrong the first time, make sure you get it right the second time. Because I will repeat questions. You cannot forget anything. I have had students tell me that come about test 4, gee, the stuff in test 1 is so easy now because they read it every single time. So when you take your final, again, 70% of your final is stuff from your old tests. Okay? Then on your final, the last 30% is just being able to interpret it. If I give you these reactions, can you tell me what this is? Okay? So it's kind of reverse of what I'm asking you now. So genotypes and phenotypes of offspring, if I give you the genotypes of parents. Why do you use Dilocus biflorus? What does it separate? Dilocus biflorus, we use that for which separation? A1 versus A2? And Eulexis europus, which one separates that? Your H versus your little h, right? And that's test 2. Okay, so you're gonna go to lab, and I do not have any fresh blood specimens, I just have what I have for your kid. So, yes, ma'am.
There's actually, at least one from